Welcome everybody to Wall Street Reporter's next super stock company spotlight. This is where we bring you those stocks which have that 10x to 100x upside potential. These are companies going after massive multi-billion-dollar market opportunities that are at a key inflection point and that have multiple catalysts in place. So today we are back with Mentis Life Sciences. Stock symbol is NMLSF, and we have Dr. Marvin Hausmann. He's the Chief Medical Officer. And we're going to be talking about their psilocybin-based therapeutic that's going into the clinical trial for autism, Fragile X, and with a special guest, Andrew Charrette. He's the Director of Regulatory Affairs for KGK Science, and that's the clinical research organization that's going to be running the clinical trial. Andrew, Marvin, welcome.
Thank you, Jack. Thank you for having us.
Thanks, Jack.
Yeah, Andrew, so it's great to have you on for the first time. What I'd like to do is, I want to first start off with, a little bit of a background for anybody that's new. We're going to talk about why, Nova is getting why they're developing this drug. But first, Marvin, I really want to acknowledge that, this is really a major milestone. I mean, we're really at a pivotal moment right now for Mentis. This is what we've all been waiting for. I mean, I've been waiting for a year. Some people may have been waiting a long time, but this is a key milestone for any company that they're going from preclinical to actually going into human clinical trials, this phase 2A clinical trial. Last time we had you on, we were waiting for the approval from Health Canada to start the trial. Now we finally got it. Everything is out of the way. We have pharmaceuticals manufactured. So it's a major milestone. So congratulations.
Thank you. No, we've waited. I've been waiting about 3 years for this. It takes time. I've done a lot of drug development in my life, and if it's done properly, it takes time. You just can't shortchange the production of a new pharmaceutical ingredient and getting the regulatory agencies to allow a study. It's a great moment. It's exciting.
It is, and here's the thing. I think for investors, what investors really need to understand is a lot of companies in the space. There's a lot of biotech companies, and especially in the psychedelic sector, which you're part of. There's a lot of companies that make all sorts of promises, and then they never deliver, and they never get anywhere even close to a clinical trial. And here you are. You've actually, the company has delivered on everything. Every milestone has come into has happened. Now. Sometimes it takes a little bit longer. Sometimes it takes a little bit longer than we expect, but here we are. We're ready to go. So, Marvin, I want to first start off with, again, for anybody that's brand new, tell us about, Mentis's the program, the drug that you're developing for treating autism, the psilocybin-based therapeutic.
Well, I was retired. I was approached about 3 years ago by the founders of Pills Bioscience, which is subsequently acquired by Mentis, and they wanted me to develop psilocybin because I had a lot of experience developing compounds from mushrooms. I told them I'd be very interested in joining them if I could do psilocybin in autism, because I just feel autism is a devastating phenomenon. It's a problem for the patient, it's a problem for the family, it's a problem for society. And I wanted to specifically focus on autism because my own theory was a neuroinflammatory disorder. And being a neuroinflammatory disorder, I envisioned psilocybin to be a perfect compound as a serotonin agonist and also as an anti-inflammatory agent to work in this particular disorder. And it was called the spectrum, a disorder, autism spectrum disorder, which means that it's a wastebasket. You kind of throw, if you don't know the diagnosis, it's autism. And we had that with autoimmune disorders when I was in medical school a long time ago. And if you didn't know what the diagnosis is, it was an autoimmune disorder. Now, how many specific autoimmune disorders we have? Scleroderma, systemic lupus erythematosus. It goes on and on and on. And I think the same thing's going to happen in autism. And it's exciting to me to be involved here. And we've accomplished what we started off. It's a great moment to have a phase 2A study approved by a regulatory agency, and we're waiting to recruit our first patient. Could occur any day now, could occur by 5 o'clock today, who knows? So we'll see.
Right. And again, from the way I look at the company, kind of from the investor standpoint, is this is a key milestone, which becomes it's a valuation milestone, right? So now companies that are developing novel therapeutics and psychedelics that are in phase 2, we've seen companies trading at valuations of 100 million plus, right? Next, Nova Mentis is trading at a valuation of under 10 million, I think 8 million, 5 million, somewhere in that range. So there's a real gap that could be closed, especially once this clinical trial gets underway, especially if we get some positive data. Now, with that said,
we get no respect. We have no respect here. We will get respect with positive data. There's no question about it. It's quite a dramatic increase from 5 million to 100 million. God, from your mouth to God's ears, Jack.
Well, no, again, I'm not making any price forecast or any type of, as I say, price representation. I'm just talking about.
I'm going to drink to that, Jack. I'm going to drink to that right now. I'm going to have a drink for that.
Well, Mark, but like in the comps in the space, right? Other biotech companies that are in phase 2, especially the psychedelics. We could mention a whole bunch of names, but some of these guys are trading at multi-$100 million valuations that are, relatively speaking, the same kind of level where you are. So I think
Jack, you know that I've been there before. I had a New York Stock Exchange Corporation. We went from $2.50 a share till we were bought out by King Pharmaceuticals at $38 or $40 a share, from a valuation of almost $500 million in 1998. I've developed drugs. I know what's involved, and it's an exciting time. And public beware. Watch what happens.
Exactly. So this is the thing. Again, from the investor standpoint, what I like about this, the whole investment story is you're going after this massive opportunity. The team, which you're leading the medical team. I mean, there's a massive track record. I mean, you've taken drugs through the whole regulatory process onto the market. You've had wins in the market. The chairman, the CEO, everybody's had a really strong track record. And then we have Andrew. Andrew, now let's turn to KGK Science. So you're the clinical research organization that's going to be running the trial, right?
That's right. Yeah.
So for anybody that's kind of new, again, a lot of people in our audience, they're not really biotech people. So explain to us what your role is in terms of running the trial, and what are going to be some of the milestones of things that we have to keep an eye on.
Sure. So KGK is acting as a third-party tester for Nova Mentis. So we've partnered with Nova. They've hired us to design the clinical trial and basically run it through from start to finish. So getting regulatory approvals, getting research ethics approvals, doing the recruitment and actually conducting the trial at our clinic in London, Ontario. And then from there doing the QA on the data, locking the database, and doing the stats and the analysis and reporting from there out. So KGK, we're truly invested in this project. We've been working with Marvin and his team on it from the beginning. We've designed a really great protocol for the study. This is a first sort of a pilot study in this indication, the first study looking at Fragile X with psilocybin. So it's really a pioneering study, and we've designed the protocol to be very comprehensive in terms of the different elements of the potential symptoms that we're looking at. So in that way, we're casting basically a very wide net in order to make sure that we are going to pick up any changes that may occur in any element of either the behavior or cognitive symptoms that we're intending to treat.
I think it's again, Marvin, maybe you can add to this. I think it's really important, from my understanding, is in terms of the way these clinical studies are designed, kind of like the endpoints, whatever the outcome you're looking for. designing the study is extremely important in order to get the result that's going to be, acceptable for regulators. Is that right?
Well, yeah, that's very important. I mean, we designed the study with all the parameters, and Andrew can address them because we had fantastic data in our preclinical. And you could ask, what led to this? It led to very robust preclinical data. We modulated behavioral changes, cognition problems in the preclinical animal, the rat, and they were so significant that we decided to go straight to a phase 2A study, and it was accepted. And Andrew can tell you more about what are the parameters and testing that we're going to be used here. But I was blown away by the finding that our oral compound in microdose therapy in the Fragile X animal was more robust, the changes, than the injectable form of the compound, which I've never seen before. Usually when you give an injection, you give a full bolus, it has its effect, but in this case, we fed it. It goes through the esophagus, through the stomach, into the small intestine, and the microdose therapy worked perfectly, even better than the injectable, which is hard to envision.
Yes, okay. It's a massive distinction. Again, if you can get an oral pill, people would prefer to take a pill, a capsule, rather than getting an injection. I mean, it's a complete world of difference. I mean, the cost, everything it's like night and day.
Well, you have an autistic child. I mean, autistic children have behavioral anxiety issues.
Yeah.
I think oral will be a lot better. In fact, you can break open a capsule, throw it into a smoothie, throw it into whatever drink the child wants. What I was trying to emphasize, though, besides the oral, was that we had a memory effect that was robust. We may be sitting on a very unusual compound in terms of being an agonist of serotonin in the brain, that we may have a normal memory drug here. Who knows? We're going to find out.
Okay, so that's, we've touched on this before. I think we're going to probably talk about it later. Today, I really want to focus on the clinical trial, but the other aspects, the other possibilities that you can have, especially, let's assume we get this, through the approval process, we get into commercialization. At that point, then you could expand, you could have label expansion, you could have off-label use for potentially other things, like talk about like this as a memory drug. I think Marvin, last time, one of the first times we were on, you were talking about the possibilities of having some beneficial effects for like other neurological disorders like Parkinson's, Alzheimer's, things like that.
Yeah, that's true. What happens, and it's happened to me before in my New York Stock Exchange Corporation, that you have a small focus of approval. Doctors can use the product in off-label use. You can't advertise it. You can't expose the product. The company can't say this is where we recommend. But a doctor in his office has a patient, and he can decide if he wants to use this in Parkinson's disease, he can do that. And then if a doctor wants to do his own study, he can do a sympathetic investigator-initiated IND with the government. So we're sitting in a nice position. But the bottom line is you need to have positive results.
Right.
You have to show in statistical significance that we're having an effect on Fragile X syndrome. That is the key. That's the key to the whole enchilada right now. This is what we've worked for, and this is what we're about to evaluate. And I can't wait till we admit that first patient. It'll be a blessing come true.
Yeah, but again, the big picture is, I mean, the possibilities for this psilocybin microdose therapeutic as a treatment from other neurological disorders beyond autism is enormous. But first, we got to start with this. Again, the road to 1,000-mile journey begins with the first footsteps, and now Nova is clearly on the path. Andrew, I want to ask you, Can you tell us a little bit about some of the details of the clinical trial? Maybe, I don't know if you want to go into, can we go into like, yeah, how the trial is designed? Like what's the, what's the endpoint? What kind of news flow we can expect in terms of milestones? Like, are you going to announce, like what kind of announcements can we expect to see? Because I think that's what investors care about. Like, are we going to get info on patient enrollment? Are we going to get any type of news, based on some initial data? I mean, look, tell us the details.
Absolutely, absolutely. So we've been at the point of seeking research ethics approval at right now, and basically we've received the approval. We haven't press released it yet. It's probably coming later this week. We've received the research ethics approval. We're just waiting for them to issue a couple of the documents that they need to finalize on their end. And what that means is that we can start screening patients, which is a major milestone for the project, right? We've gone through the regulatory approval. We gained not only the approval for the first trial in Fragile X with psilocybin, but one of the first microdose studies in which the product could be taken home by participants. That's a big piece for psilocybin, as it's
okay. That's an important distinction. I don't think anybody is aware. I was not aware of this. What does it mean? So in other words, other
right. So because psilocybin is a controlled substance, most trials are administering it in the clinic.
Okay.
And not allowing patients to take the product home with them. And what we gained approval for was to actually send the product home with the patients and their caregiver, take the product at home in more of a real world setting, which is big. There's one other trial, I believe, in Canada that gained approval for that just ahead of us, but that's a very big thing, because it really will show, or the practicality of microdosing psilocybin at home, which is a big thing. And in terms of the trial itself, it's a small open label study, and but it's very important for exactly what Marvin said. We need to assess whether the product is working, and one of the main or the primary endpoint of the study is feasibility, and the reason for that is because it is the population that we're studying. So we want to make sure ahead of assessing the efficacy and the effectiveness of the drug, are we studying this the right way? Are people completing this study? Is this something that treatment something that they can do? Is it feasible? Now, the pieces that we're interested in the most are, of course, the efficacy endpoints, which we're looking at several different markers of cognitive function. So Fragile X syndrome patients are developmentally delayed. So their IQ for the trial needs to be between 40 and 80. So that's a significant deficit in IQ. And what the preclinical data shows is that there's significant changes in cognition in the rats. So we're hoping to see that carry over in the study. So we're doing a lot of sort of the classical tests for cognition. We're using the NIH cognitive battery, NIH toolbox cognitive battery for intellectual disabilities, doing a trail making test, which is a classic test for switching attention. We have an executive function test that's called the Brief A, another very widely used test. And then we're also assessing a lot of behavioral markers, because about 40% of people who have Fragile X syndrome have autistic symptoms. And so we want to see how the behaviors are affected. So we're looking at aberrant behaviors, the repetitive behavior scale, looking at the multifaceted empathy test, which is a very important one for autism, and then the Vineland Adaptive Behavior Scale, which is another very widely used test for autistic symptoms. So we have, and then we're assessing some other elements as well. So anxiety and mood, we're using a test for that as well, which, you know, psilocybin is being investigated for depression and anxiety and in other indications. For people with major depression or general anxiety, we're also going to look at those mood symptoms in this population as well. So it's very comprehensive in terms of the fine elements of cognition and behavior that we're looking at throughout the study. So I kind of jumped right into the endpoints, but the trial is 30 days, so people will be taking the product every other day for the 30-day duration, and we'll be testing them weekly on several of these endpoints.
Okay, now question for you. Now, just to be clear, are you how many patients are going to be in the trial?
So there'll be 10.
There'll be 10. Okay. Now, do you have to wait to get all 10, or can you start as you get them? You can start this as a rolling.
Yes, it'll be rolling recruitment. So in terms of announcements, as soon as we enroll the first patient, that's typically something that we'll announce. And from there, we expect recruitment to proceed fairly quickly because it is only 10 folks. It's really, you know, it's a very specialized population, so it's not going to be as straightforward as.
Right. You have, I mean, you have already, you have orphan drug designation from the FDA because it's a very niche. I mean, it's, you know, how many, I mean, how difficult is it to get the patient, you know, the potential patients into this trial? Because, you know, Fragile X is kind of, it's a niche. I don't know if that's the right word, but it's kind of a niche area. I mean, it's an orphan. Yeah, that's why you have an orphan drug designation.
Yeah, so I mean, it's between 1 in 4,000 to 1 in 8,000 people have this disorder. So it is rare, but it's only 10 people. And to be honest, the Fragile X community, we've been in touch with them. They're very engaged, very excited about the trial because there's very few options for them in terms of treatment. There's no treatment, no cure. It's a genetic disorder, but even for symptoms and treating symptoms, it's very limited. So I think the Fragile X community, the caregivers are a big part of this study, I should mention as well. They have to agree to participate. They have to answer like a good portion of the questionnaires that we're asking are completed by the caregivers as well. So everyone is very engaged. So I think recruitment will go quite smoothly. People are very interested in anything that may help.
Yeah, of course. Now, so in this study, are you going to? So will you get data on efficacy, or you will get efficacy? Okay.
Yeah. So the way that, you know, Health Canada likes to see early studies look at feasibility and then look as the primary, and then secondary look at efficacy and safety and that kind of thing. So that's the way that we've set it up. But it really sets up the trial to assess everything we like, and from here, further studies will be very well informed on how based on the results of this trial.
Okay, so earlier you were talking about, I think you just gave kind of a high level overview of some of the different level of approval. I mean, because there's a lot of paperwork involved there. I know, like, it wasn't just the Health Canada approval. I mean, to get that, there was a whole bunch of data you have to get, and then you have to get controlled substance approval because it's a psilocybin that you have. I mean, how many approvals do you need? I mean, I think now there's another one that you're doing, which is like, I guess, like the last one, which is, I mean, like sort of a formal, it's a more or less a, but this is, so it's an involved process. Okay, so bottom line is, I guess what people want to know is, for the layman who's watching this, they want to know, okay, how long is this going to take, this trial, and when if they get good data, and what is good data, like exactly what is going to be, in other words, what kind of data? I think what everyone wants to know is, what is the type of data that the market would be excited about that says, hey, this thing has a shot at commercialization. It could really address this massive unmet need, and it could get into, it could get approval at some point, get an FDA approval. Talk about that, please.
Sure. So, so I guess if we see statistically significant changes in any of those markers of efficacy in terms of behavioral or cognitive function, it's something to get excited about. This is there is likely going to be studies to follow up on this, and whether that's to seek the orphan drug. The beauty of Marvin's strategy and Nova Mentis putting this through the orphan drug route, and this indication in particular, is that it has implications in cognition, not only for Fragile X, which is, as you mentioned, a major unmet need, but there's also potential implications in cognition and in autism, right? So if we see, with this first study, if we see positive effects in any of the behavioral cognitive symptom or questionnaires that we're asking about, then it's something to get excited about, not only for Fragile X, but for some of these other potential indications. And that's where having such an extensive battery included in this study really positions us well to pick up changes as they may come up.
Okay, so I'm not that at a high level guy. I'm getting the idea that you've given a lot of information that somebody who's a professional, like a biotech pro, they can kind of read between the lines. They can see, hey, this could be something really big, which goes far beyond just autism from this study. I mean, this, in other words, like somebody who's
the formula of life is very similar, no matter whether you have Parkinson's disease or Alzheimer's disease or autism. These are neuroinflammatory disorders. And remember, cognition or memory is just a symptom of that disorder. Dementia is not a disease, it's a symptom of the disorder. Dementia is in even MS, multiple sclerosis, Lou Gehrig's disease, Huntington's chorea. Even diabetics who have neuropathy can have dementia. So the formula, if it works here, it's a large area in medicine, and it's up to the doctor to interpret what he sees from our study. And I just want to go on record. These guys at KGK are top drawer. They did a beautiful job for us, and it's a great relationship, and it's just beginning. It's an exciting relationship, and I can't wait to admit our first patient. It'll occur any day now, and Jack, you welcome to interview us again after we have the first patient.
No, 100%. Actually, Andrew, I want to ask you. So, for anybody that's kind of not familiar with KGK, can you talk a little bit about like other companies you've worked with? Maybe talk about some success stories, other drugs you've helped get through the approval process, like so people understand what exactly you guys do.
Absolutely. So KGK is a contract research organization. We've been around for 25 years. We are a wholly owned subsidiary of Wellbeing Digital Sciences, which is a publicly traded company on the Canadian, on the NEO stock exchange up in Canada. And we have a focus, our company, KGK Science, has a focus on conducting clinical trials for nutraceuticals, cannabis, and for psychedelics. And we also provide regulatory consulting and regulatory services for those industries as well. So over the 25 years, we've had, if you go into a Walgreens or a CVS, there'll be a lot of products on the shelf in the dietary supplement space for which we've done clinical research or helped them with some of the regulatory filings. On the psychedelic side, I can't talk about all the companies that we've worked with, but we're currently helping a company called Hallucinex Life Sciences with their phase 2 clinical trial of psilocybin. We've worked with another company called Psych Wellness, which is also a publicly traded company. There's been, our relationship with them is public as well. We help them from sort of the inception of their Amanita muscaria extract up through all the preclinical and phases, and they're now sort of entering the market in the US with that extract, and they're looking at clinicals as well. I think I'm not sure, but yeah, we've helped several companies already in the psychedelic space, and yeah, there's 25 years of success, I guess, on the nutraceutical side as well.
Okay, so the data. Okay, so the phase 2A clinical trial is about to start. The data that you're going to be getting from this trial, like this is, are you getting any? I think Marvin, I think you mentioned like there were some DNA markers that you were looking for also that were like absolute.
Yeah, DNA is fixed. Yeah, I'm sorry, Jack. DNA is fixed. The translation of DNA is through mRNA, messenger RNA. So Nova has a patented technology that it developed with Thermo Fisher Scientific, the big genetics company, using about 52 mRNA genes. And these genes produce cytokines or inflammatory markers. And we're envisioning using these, and as part of the study that's been approved by Health Canada, this is a tertiary outcome. Your primary, secondary outcomes are your behavioral and cognition testing that Andrew addressed earlier. We've also got approval for exploratory outcomes. That means that we can use them or not use them depending how they turn out. So we have two assays we're using. One is the mRNA cheek swab. It's non-invasive. You don't have to draw blood. Just use a Q-tip or a special Copan swab. It's a little more sophisticated than a Q-tip. It accumulates the cells on the cheek, and then we can extract the RNA. We developed that technology. You have to extract about between 20 and 40 nanograms to be able to measure it. This is not like a big molecule for DNA. And the other test we developed is with called the serotonin assay in saliva. So there are exosomes, these small vesicles that penetrate the blood-brain barrier. They seem to accumulate, not seem, they do accumulate in the saliva, and they carry these inflammatory markers, cytokines. They also carry serotonin. So the serotonin that's produced in the brain travels through by the exosomes, through the blood-brain barrier into the saliva. So we have made a big breakthrough with University of Central Florida with Dr. Kimi Sugaya, who's the head of neuroscience there, and he has been able to develop the antibody to measure serotonin in the exosomes in saliva. So we're very excited, and that's going to be very unusual data. If that works, then big pharma is going to want to look at this because they need this data in depression or any drugs. Biogen is developing antidepressive drugs. They're developing an MS drug. This could be used to evaluate therapeutic response.
Would this be a separate product and this mRNA analytical data? This is separate from the psilocybin?
Oh, absolutely. It could be used elsewhere, but no, it's part and parcel of the phase 2A study. But that data bank is applicable. If that works, that data bank could be used in other drug development. I remember when I had Medco research, we developed Wellbutrin, which is now one of the largest selling antidepressants in the world. So I know the industry well. This data is applicable to other neuroinflammatory disorders.
Okay, so my understanding is that with this mRNA data, so essentially this gives you, Andrew, maybe you could speak to this, that in other words, this it's absolute. It's not really subjective like some of the other data, the behavioral data you get, it's kind of subjective, but this will kind of give you some real absolutes onto whatever it is that you're looking for. Is that right?
Yeah, remember, any time you're treating a patient, you have a placebo effect. You have the parents want the child to be better, the guardian wants to be better, the child wants to be better. So hard biomarkers, in fact, the FDA has what they call real-world evidence and real-world data. And I'm going to give you an example. I just got called by someone, I can't go into details, by a very large pharmaceutical company, because Biogen just had a clinical advisory meeting, and I forgot the drug. It's called tofersen, I think, and it's used in I think multiple sclerosis, and the drug was not better than the placebo in terms of symptoms. The Clinical Advisory Committee has recommended approval based on the fact that the drug lowered a neurofilament, a toxic neurofilament that is present in multiple sclerosis. That's probably one of the causative toxic proteins. Remember, so the symptoms were the same as placebo, but the fact that there was a significant, statistically significant lowering of this neurofilament, the Clinical Advisory Committee has recommended approval. Now, whether the FDA goes along with that, we don't know, but it's showing you the significance of some of these hard inflammatory biomarkers. And the same thing in Alzheimer's disease. They just discovered a soluble protein that's present in people who have died of Alzheimer's disease, and now they're finding it in normal people. So is this a predictive test for those who are going to develop Alzheimer's disease? So this whole industry is just taking off.
Okay. No, it's very interesting. So there is, I mean, so really there's a lot of possibilities for, what Nova is developing, a lot of market, opportunity for, sort of Nova's.
I will say that the biomarker piece is a very exciting element of this study, because I think, as Marvin said, the objective biomarkers really support the sort of subjective questionnaire, as you mentioned, Jack. And that's a major piece that's missing in psychedelic studies thus far. There hasn't been an in-depth look at biomarkers.
Nobody has done biomarkers in psychedelics. Nobody has given biomarker data in psychedelics.
There is some, but there needs to be more, and that's a huge element of this. And some of the biomarkers that that Nova has developed are, they're looking directly at the chemicals in the brain that you cannot extract any other way, which is very novel. You can do imaging of the brain, but you can't, without taking a biopsy, you can't look at changes as some of these things in the brain. And that's why it's very exciting what they're doing. So it has implications not only for this trial, but across all sorts of mental health disorders.
Okay. So,
yeah, if you think of what I just told you, I shouldn't go there, but take Biogen, for example. It could be any other company. In order to diagnose this neurofilament change, you may need a spinal tap. But if we have an mRNA panel that could show the changes in this neurofilament, it's a home run.
Okay. So back to the clinical trial. So we're essentially waiting for this one last piece of approval, which is more like a formality, I guess. This week, maybe you might get this, and then you're going to start recruiting patients.
Yeah, exactly.
Okay. And can you talk about like news flow? Are you, Marvin? Maybe you can add to this, because you're with the company. Andrew's a separate organization. Would we be able to see news like every week or so, like giving us kind of updates on, okay, we have one patient, two patients, we have this data, we have that, what kind of news flow can we get? Can we expect?
Well, I can't address that right now. I mean, we want to first recruit our patients, and then we'll make that decision as we go through the study, how much we want to reveal to the public. I mean, remember, you have HIPAA rules. You got to really be careful. We're treating human beings who are sick. And so we have to have it with dignity, and we have to treat the whole study as a dignified study. And we have ethical approval through institutional review boards. We just got that. And so we're going to tread softly to see how we want to reveal the results from this study.
Okay, fair enough. Okay, and timeline, like when, so let's say, how long do you think it'll take? I think I asked before, how long would it take to get these 10 patients? Do you think, can you say it? I mean, to get the 10 patients, do you have ballpark?
I'll let Andrew address that. Andrew, I'll let him.
Sure. Yeah, it is hard to say because, as I said, we have a lot of interest. We're hoping that it's a matter of a few months or less, because there is quite a great level of engagement. But until we start actually screening the folks, it's hard to know because they need to have a certain IQ, they need to have certain, they can't have certain medical diagnoses or mental health diagnoses on top of the Fragile X diagnosis. So there's a lot of things that could weed people out along the way. Now we've just kept it pretty open, and we're hopeful that it'll move quick, but it's difficult. It's always difficult. Recruitment's the hardest part to predict, but we're hopeful that it's done within a few months.
Okay, great. Marvin, back to let's talk a little bit in this. Okay, so in the psychedelic sector, developing different therapeutics, you're using a microdose, right? Like I think others are using a macrodose. So why micro with psilocybin versus macro, and what's the difference for anybody?
Well, it's quite a difference. The macrodose has hallucinogenic side effects, and a lot of companies are developing macrodose psilocybin for depression, treatment-resistant depression, cancer, depression associated with cancer. To me, I didn't envision giving a child with autism a dose that has a hallucinogenic side effect or other side effects. And from my experience in drug development, there's receptor activity, there's binding of the product you're developing to a receptor. In this case, it's the 5-HT2A receptor in the brain. Serotonin binds to that receptor. We also know that when an autistic child after birth has a much lower level of serotonin than a child who doesn't have autism. And then as the child grows up, the child who doesn't have autism, I like to call everyone is normal. I don't want to say abnormal. The child that doesn't have autism, the serotonin starts going down. The autistic child, the serotonin at age 10 goes up. So it's about 200% difference at birth and 200% difference at age 10. Now, psilocybin, I hope I'm not getting too scientific, is a serotonin agonist. That means it mimics serotonin at the 5-HT2A receptor. So it's very exciting for us to have microdose therapy, 1.5 milligrams, no major side effects, repetitive binding to the receptor. Now, when you might want to interview me in the near future, because what's the logical next step in research? If you have a serotonin agonist called psilocybin, if I block the receptor, will we still have positive data with Fragile X syndrome in the animal model? Which would be really very interesting. So if I block that receptor and I still have a dramatic symptom change in the Fragile X, I have a whole new mechanism of action for psilocybin, and the industry might be very interested with these results. Those are called pharmacokinetic studies. And I don't know anyone who has done, unless Andrew knows someone, that has blocked the receptor and has shown microdose changes with psilocybin therapy.
I don't know any.
Wait, so how do you get that? Is that with your formulation, or how would you get this type of result?
No, you don't change our formulation. Psilocybin is a 1.5 milligram dose in humans, and it's much less in the animal, of course, because the animal is much more sensitive to psilocybin than a child. And so that's how we came to a 1.5 milligram. It's about 0.1 milligram in the rat. It's a much smaller animal, of course. So psilocybin's not changing here. The only thing we're doing is blocking the receptor. So here comes psilocybin, it's coming along, it can't bind, but it still has a dramatic effect in the disease. And that's what we're working on now. So stay tuned, Jack, for the next interview.
Okay. Let me see. Let me pull up my notes. A couple of things I want to cover here.
I hope I didn't lose the audience on too much science there, Jack.
The binding stuff is a little bit. Okay. Marvin, look, can you talk about like the capsules? You know, what's the final drug formulation? You know, where are the capsules being produced?
Well, the product was produced by a large manufacturer in the United States, and then we identified in Canada the Toronto Institute of Pharmaceutical Technology, which is part of the University of Toronto. And they have an excellent formulation division that can produce capsules. Remember, just having the drug as an API, active pharmaceutical ingredient, is not ready for big time application in the human. You have to think about stability studies. You have to say, are you going to give it by injection? You're going to give it by capsule? You're going to give it by a compressive pill? We chose capsule. So we could put 1.5 milligrams exactly in the capsule, and the capsule allows the guardian or the parent to open it up and put it into a drink as well.
Okay.
We needed an excellent organization. And TIPT, or Toronto Institute of Pharmaceutical Technology, and by the way, they were recommended by Andrew. That's why it's a great relationship with KGK. And we've established a superb professional relationship with TIPT. And so we have more than enough capsules for the study, and we could go on and even sell it commercially if we were allowed to, but one step at a time. So we're producing that now. They're ready to go. They'll be in blister packs, and the parent can take them home with the guardian, and they can administer it to the child in the home setting.
Okay. And Marvin, what about, do you have any other preclinical studies, any other data that you're getting? Because I know like you have studies going on all the time. Anything else since last?
Well, I did tell you about the pharmacokinetics. I'm not at liberty really to go into that right now, but I'm saying stay tuned, okay? Those experiments are ongoing as we speak, and we're very excited to see what the results are.
Okay, so bottom line is,
by the way, those studies, sorry to interrupt, those studies are going to be with Dr. Treza in Italy.
Okay.
She's the one who has the superb models for autism in the rat. So we're using the same model to look at the pharmacokinetics of psilocybin in autism.
Okay, so the bottom line is we've got the clinical trial, which is could be coming up very soon, where the patient recruiting is about to start, like literally, you know, could be a week, 2 weeks away. That so we get those announcements, and then the actual trial starts, and then we could have data coming in. I mean, I'm being optimistic. Is it possible that we could see data? We're already in April. Could we see data by end of the summer? Andrew, Marvin?
Yeah, I mean, the study's only a month long, right? So depends how fast. It's the recruitment piece, as I mentioned. We're well positioned for it to go quickly, but I don't want to say it's going to take this long because we never know. It's, as Marvin said, it's human beings, and there's a lot of variables there, so it can be tricky. But it is possible that by the fall that there's data coming in. Yeah.
Okay. So just to be clear,
Jack, as much as you're excited, we're excited. I can't wait for the. I mean, this is a great. We've waited a long time. We're now knocking at the door, Jack. We're knocking at the door, so it's exciting.
Yeah, and again, this is so to be clear, the trial is only 30 days. Kind of the longest part of it is getting the patients into recruiting the patients. But once we have that, it'll, you know, it's 30 days. It's not something that's gonna, you know, drag on for 2 years. It's basically, the whole process could take, in theory, let's call it a few months. Let's call it 3 months potentially. I'm saying 2 months to recruit, 1 month for the data. No, it's 3 months.
Yeah, but Jack, you have to keep one thing in mind. Remember, you got to do statistical analysis on the data. Just because you have symptom improvement in the child and the parents and the guardian are excited, we have to show hard statistical evaluation of all of these testing procedures.
Mm-hmm.
That takes time.
Validation of the study data, and then the stats. So that does take time, usually a couple of months, 3 months, but we'll be able to know, the data's come in, and there might be some top line comments shortly after that.
Okay.
Just depends how things go. So.
Okay, top line comments. Explain what would that mean?
Well, they may just say that.
You put your foot in it, Andrew. Here you go.
Okay, so
I interrupted Andrew. How would you define top line, Andrew?
It really depends. I mean, when you look at it's usually something like one of the major endpoints. You may just pick one endpoint to look at to say, it was we saw positive results or not, and maybe not numbers, but maybe just a comment on, whether or not positive results were obtained or not, something like that. Now that again could be very. It depends on how quickly things move, and the statistics can take a while. So it's hard to say when that would be, but that's usually something that you could look at first if you need, if you're wanting to let investors know how things went.
Oh, okay. So in other words, it's possible to release top line comments in middle of the trial. Can you do that, or
not in the middle, at the end, but before all of the detailed work has been done.
Okay. So it's possible. I mean, there's a potential. If things go well, there might be potentially positive top line comments. We're not saying, we don't know, but it could be, and that could be okay. So the bottom, Andrew, I'm just trying to figure out what are the catalysts that could move the stock. That's kind of what I'm looking at. Like, okay, so this is one thing, and that could be, potentially a few months away. Let's say you get the recruitment in, you get the trial completed. So you may have a catalyst in about 3 months potentially, and then when you have the actual statistical data, that's going to be the real that's the real make it or break it moment.
Absolutely, yeah.
Okay.
Yeah, we could identify when we're submitting. So we could say that we've accumulated the data. We're now submitting the data to Health Canada. I mean, many companies make that statement.
Okay. And what happens? Let's say you get the endpoints you're looking for. I mean, let's say things are reasonably positive, right? What's next? What happens after that? Like walk us through the path towards potential commercialization.
Well, Jack, I mean, we're going too far afield now. I mean, the first thing, let's walk before we run. The first thing we have to do is see how robust the data is. I don't mean to avoid answering your questions, but it's not possible to answer right now. We have to see how robust the data is, what the statistical data and significance looks like, and then we can make a decision. What are the next steps? Do we have to repeat the study? Can we go and push for approval? Can you get sympathetic approval? Remember, this is a need. This is an unmet medical need. There is nothing that I'm aware of in Fragile X syndrome. And how we deal with it and how Health Canada deals with it is an unknown, and it just can't be addressed right now. Let's start the study, Jack.
Okay, very exciting. So the bottom line is we're at another milestone. This is, again, it's a big deal for any biotech company, especially in the psychedelic space, that we're actually going to get to potentially this, a repricing moment. If we get the good data, the stock gets potentially very positively repriced. Okay, gentlemen, thank you, and I'm looking forward to maybe a follow-up. When do you? I guess maybe we should do a follow-up once you have the first couple of patients enrolled. No?
Yeah, absolutely. And I want to thank Andrew for coming on with us. I mean, you're giving
Yeah,
a different perspective, a
absolutely.
regulatory perspective. And I also wanted to tell you, we're blessed to have KGK and Andrew and how we're doing this whole program. You can't believe how many hours have gone into this, and to have experts of their caliber on your side means everything. So I want to thank Andrew and his team for helping us.
Thank you, Marvin. Thank you very much.
Andrew, thank you again. Marvin, thank you. Hopefully we'll catch up with you guys in the next, let's hope, hopefully in 30 days, potentially 30 days for, the trial update.
Thank you,
again.
Chad.